But I'm really extremely excited to introduce Dr. Brooks. Dr. Brooks is coming from the NIH, which you can imagine wasn't an easy matter to arrange, so I'm grateful to him for jumping through those hoops. He's the Deputy Director of the Division of Rare Diseases at the NIH, and I think the Director of the Bespoke Gene Therapy Institute still? Yeah.
One of the co-chairs.
Good, one of the co-chairs. So Dr. Brooks is going to talk to us about therapeutic platforms, rare monogenic disease, and clinical trials. So without further ado, please welcome Dr. Brooks.
Great, thanks Hans and Surge Leadership for the opportunity to speak here. I've never spoken at this meeting, but very much looking forward to it. I work for the NIH. I don't have any financial conflicts of interest, and anything I say are my own views and not those of HHS or NIH. So when we come into this, and sort of as a background for all I want to say is the ever-increasing number of rare diseases with a known molecular basis, which is now estimated to be about greater than 10,000 or so, and increasing with the increasing availability of whole genome sequencing. But the number with approved therapies is still around 600 and lagging far behind. And the first director of NCATS, Chris Austin, did a calculation. He said that at this rate, it would take about 2,000 years to develop treatments for every rare disease, which is a good and memorable line, but it misses an important point, which is it makes the assumption that anybody's actually going to develop treatments for all of these diseases. And in reality, amongst rare diseases, some are a whole lot rarer than others. And this was taken from a paper published by our European colleagues in OrphanNet, and they're looking at the different prevalences of different rare diseases. And in this sample they looked at, there's about 150 or so that have high prevalence and obviously high commercial interest. But at the other end are the 1 in a millions, and some of the ones that I started working on in a lab many years ago, which are of almost no commercial interest. And to understand the distinction here and the importance, we think about, that we've got an amazing gene therapy, Zolgensma for spinal muscular atrophy, right? Kids would be dying at the age of 2, now they're walking around. SMA affects about 50,000 patients in the United States. What would have happened if everything was the same, the science was the same, but there was only 50 SMA patients? They'd still be dying at the age of 2. And it's understandable, right? For-profit companies are for-profit companies. And I say this not to be in any way critical of big pharma companies. Indeed, like many people of my vintage, shall we say, every morning I wake up and I take 4 or 5 little pills for the typical age-related maladies, and they work very well. They're all made at big pharma. Thank you, big pharma. So that isn't my point to criticize, but to understand the reality of the situation. And that if we're going to ever address all these diseases down here, we need, in my view, a fairly radically, fundamentally different approach. And what I'll talk to you about is some thoughts about what that kind of approach might be. So, there are lumpers and splitters in the world. When it comes to rare diseases, I am a big lumper. I want to lump things as much as possible around potential therapeutic implications. So if you think about monogenic disease, of which there are thousands, when you get really right down to it, the underlying etiologies that we're trying to treat, there's a very small number of those, way smaller than the number of diseases. So you think about things like, I think of these as sort of the big 4 of the monogenic diseases: nonsense mutations, for which you can have stop codon read-through drugs. I think we'll be hearing about that. Missense mutations, abnormal protein folding, abnormal RNA splicing, we have antisense oligonucleotides, and for dominant disease we have gain of function mutations and siRNA therapies for those. And you can also break it down. There's also signaling abnormalities that cut across many diseases, and there are drugs that can be used to address those. But the even bigger therapeutic approach is to get to the really fundamental problem in monogenic diseases, which is the genetic defect. And for there, we can think of either gene therapy and gene editing, which would really get to that fundamental issue, and those are the really sort of big buckets. For these, it really comes down to, if it's a monogenic disease, one of the good things about monogenic disease, in fact, is if it's a monogenic disease, if you can get a working copy of that mutant gene into enough cells at the right time in disease progression, something good should happen. You might even prevent the progression of disease. You might even prevent it all the way from the beginning. So that's kind of what we're looking at, and that's what a lot of what I'm going to talk about is about. So one of the first things that we developed at NIH is a program that we call PAGT, Platform Vector Gene Therapies. And the original idea of this one is we'll identify 4 different monogenic disorders, each of which could be potentially treated by AAV9. And if we just kept using AAV9, once we got a regulatory approval for the first one, for the second one, we might reduce the number of steps and make the whole process a lot more efficient, because we're basically reusing the same delivery system. As we move through this, for various reasons, we're going to actually use different vectors for the 2 organic acidemias and the 2 myasthenic syndromes, but the idea is the same. Can we leverage the therapeutic platform? And this is done in collaboration with our colleagues at NHGRI, Chuck Venditti and Carsten Bonneman at NINDS. And another thing that we really want to do here is, as we go through this regulatory process and interact with the FDA, we want to learn, get their feedback, and then make that information publicly available, which would be a crazy thing to do if we were a for-profit company, because we'd be giving away our intellectual property. But we're not. We're the government. And so we do want to make that information available. So as we've gone through, we have actually done that kind of at some early steps. We have got an orphan drug designation, rare pediatric designations for the first of these, which is a AAV9 for treating propionic acidemia, and published that, and more recently another publication that talks about the Interact meeting and the things that you learn from that. And these, you can go onto our website, find the actual designation that was approved, as well as a fillable form that you can use to essentially cut and paste to make your own. So these are the kind of things that we want to disseminate to the community, and I'll be talking more about this in the future. So PAGT was sort of the precursor of the Bespoke Gene Therapy Consortium, or the BGTC, that Hans referred to. And this is a public-private partnership that is organized by the Foundation for the NIH. And a key point here is that the Foundation for the NIH is not actually part of the NIH. It's a standalone 501c3 entity that allows NIH employees like myself to work with people from big pharma companies without violating any laws, essentially. They handle the money because the money has to be handled in different ways. But it's a really nice program. It's one of the multiple accelerated medicines partnership programs that they do at FNH. And as Hans mentioned, I'm the co-chair along with Tim Miller from Thermo Fisher. We also have a lot of interaction with the FDA now. Gopal Reshaduri, who contributes to that, and there's a link to the program. And this is a public-private partnership that involves multiple parts of the NIH, all contributing some money, as well as these other public entities, some big pharma, some small pharma, also some nonprofits. And roughly we end up with about a 50-50 split of the funding, the BGTC. And so when we designed this, we designed it to have sort of 2 components. One is what we think of as sort of a basic biology component to understand how, better information about the basic biology of AAV. And I should say here, all of this we're talking about adeno-associated virus, or AAV gene therapies. Can we learn about how we can enhance vector generation and or enhance therapeutic gene expression so that when it goes into the patient, it's more effective? And so those are more sort of typical research grant type programs. The largest part, though, is the clinical program. And a lot of this is focused on how can we make it most efficient to go from the concept, proof of concept studies, animal model, all the way through the ability to first-in-human trials. And we want to try to harmonize as best we can and come up with minimal packages and approaches for things like the critical quality attributes that you need to assess whether an AAV is safe to go into humans, and can we come up with a minimal set of animal toxicology studies before going into people. The goal of this is not to cut any corners or to reduce patient safety, but really to ask, what is it that we need? What is the minimum information that we need to be able to determine that a prep of AAV is safe to go into people? And so that's a big sort of focus of the program, is how do we make this whole process the most efficient and make it easiest for people to learn from us and to navigate the regulatory path. And to do this, we wanted to identify a set of diseases that would be good for this program. So we had an open sort of call for diseases, and we made different selections. We wanted some focused on the ocular system, small amounts of AAV, some focused on neurological, and some that involve systemic delivery. Multiple sulfate deficiency is one of the neurologic diseases, and that's the one we'll hear about in the Zebra and the Bear movie. And a couple other things to say about this. We were able to partner with the California Institute for Regenerative Medicine, who's funding some of these trials. When we first started, we thought we were only going to be able to do 5 or 6, but CERN was able to help us and pick up some of these. We have currently put one of these sort of, back up, kind of on hold because it wasn't making sufficient progress, and we only have 5 years for this program. We wanted to be sure we're not going to start a trial if we can't finish it. So right now we have 7 programs that are all moving towards the clinic, as indicated there. And so, as I mentioned, kind of a similar idea of what we did with PGT. We really want to disseminate a lot of information as we go to make it accessible to not only small pharma companies, but let's be honest, in many cases it is parent groups who are developing and almost having become gene therapy experts. And so we wanted to be able to make this information available. So one of the deliverables of the BGTC is this playbook, which is available now. It's in a digitized version that I think is very useful. It kind of gives a lot of the steps from going to the animal model all the way to the first in human study. Notably, it includes a lot of templates, a lot of approved regulatory documents, approved INDs, approved pre-INDs, so you can kind of see how other people have done this and make it less of a black box for those who have not done it. And as we do develop more and we get more learnings, and we indeed have had learnings from the BGTC, we will incorporate those into subsequent versions of the playbook that will remain publicly available. And so, yeah, I guess I'd just say that, and we can talk a little bit about the learnings. I think some of the learnings that we've had so far, I can tell you one that is a learning that I say I wish I didn't have to learn, which is that one of the things that limits how quickly you can get these things into trial is actually nothing to do with science. It has to do with really boring things like getting contracts signed and lawyers from one entity talking to lawyers from another entity. I wish I didn't learn that, but that is one thing that we have learned so far. But there are other sort of more scientifically relevant ones, like how do we do potency testing of AAVs in a simplified way? What are the limited numbers of animal toxicology studies we can do? And we keep kind of narrowing that down, so we will have a lot more good information here as well. So now I want to move into genome editing as opposed to AAV gene therapy, which, it's fair to say gene editing is a kind of gene therapy, but it is different. It's directly correcting the cause of mutations. And I'm going to talk about a program that was funded by the NIH Common Fund. Some of you may not be familiar with this, but the NIH Common Fund is a special bucket of money from the Office of the NIH Director for large cross-cutting projects of interest to many different parts of the NIH. It complements the missions of different institutes and centers and addresses opportunities that no single IC institute or center can address on its own, and certainly genome editing would fall into that category. So about 6 years ago or so, we started The somatic cell genome editing program, this is the first phase of that. Common fund programs can have up to 2 5-year phases, and this was the first one. So these are some of the projects that we funded in phase 1. We had a lot of focus on better ways to deliver genome editors to different cells and tissues, other than the liver, because the liver is pretty well known. Better animal models to test genome editing, unintended consequences, expanding the human genome engineering kit, and a dissemination function I'll talk about. And the goal here was to kind of lower some of the barriers to get genome editing into the clinic. And just to kind of talk about one thing, I don't know this guy. This is not part of the talk.
Sorry about that.
So we take very seriously independent replication. Oh, that would be good. Yeah, that would be good. Okay. So we had 20 delivery projects, and we wanted to be sure that as these things would get disseminated out, that they actually really work. So we put in an approach, and these are 2-phase funding awards, and the details of what we call them doesn't really matter. The point is you get one batch of funding to develop a delivery system in your lab, but before you get the next batch of funding to scale it up into large animals, we put in a requirement for independent replication, where you have to take your delivery system, send it to 1 or 2 replication centers. In mice, one was at Baylor, and one was at Jackson Labs. They had to reproduce what you got in your lab, and if they did, you were able to go on and get the second batch of money. And interestingly, of the 20 projects that we funded, 3 of them didn't make their milestones in the lab, so they didn't go on. Of the other 17 that met their milestones, 16 of 17 could be independently replicated. So that's a lot higher than people often talk about, the replication ability that pharma says that they can't replicate a lot of NIH work. This tells a different story. We can discuss perhaps why that is, but I did want to mention it. And one thing that's kind of fun, we really like this idea, was this is a paper by Guanping Gao and his team. They're looking at AAV5 to deliver genome editors into the lung. And this paper actually was published and included the people from Guanping Gao's lab and the people from Baylor who did the independent replication. There's the paper, there's the Guanping Gao's data, 20, 30% delivery. And here's the independent replication, and it was quite consistent. So we're very happy to see that both were published. And obviously we think that doing this has potential implications as shown here for the drug development enterprise in the United States. So overall we were pretty happy with SEG phase 1. We had a lot of papers. We actually contributed some of the funding to the prime editor by David Liu and others. But we really wanted to, again, make as much information as possible available. So if you go to this site, this is a toolkit from SEG phase 1. You can look at a lot of the data in here, much more of the data that was published, some of the primary data, even some of the independent replication. Again, publicly available. We'll encourage you to check that out. In the meantime, in those 5 years, genome editing continued to move pretty fast. So we were able to get another 5 years of the SCGE, and we're now into phase 2. And here we're really trying to get into the clinic, IND-enabling studies, and even a couple getting into the clinic, and a little more also on delivery. So these are some of the sort of assays. In this case, they're more developing technology and assays, but the difference here is we really want to have an emphasis on assays that are developed in a way that would meet regulatory muster. And we hope that some of these would ultimately become part of sort of the standard approach for genome editing. Ultimately, that's up to the FDA, but we really want, that was the idea we had in mind. A couple of really nice projects by Shandar Sai on better ways to detect off-target editing. We have a lot of projects, we got 5 here that are focused on really getting all the way up to an IND. They are IND enabling studies. They're all focused on either a particular disease with different approaches or a particular target tissue. Many of these are on neurologic diseases. This one actually gets into potentially prenatal therapeutic base editing. And this is an eye disease. This is, Sonia Valab is a very interesting scientist. She's trying to develop an approach for therapeutic gene editing for prion disease, and she herself does indeed have a prion disease with a disease-causing mutation. So she has a very sort of personal interest in this, and they're progressing very well. They're trying to come up with an epigenome method that would essentially shut down the gene that encodes the toxic prion. So these are fairly large projects with different approaches being tried. Again, with the idea of ultimately getting an approved IND. But then to get into, before I get into the last one, just to kind of compare something AAV versus genome editing as to what is a better therapeutic platform. So when I started with the PAGT and the BGTC, I drew up this sort of, where I thought at the time was a kind of clever diagram to explain how AAV works. It's almost like you've got these delivery boxes that look like FedEx boxes, right? That are pre-addressed to go to certain cells and tissues, and you take your therapeutic gene and you drop it into the box, and off it goes. This, I can tell you, is a gross oversimplification of the way that AAV production works. After several years in the BGTC, I can tell you that. You actually build the box, and at the same time you're building and building. But setting that aside, there's a fundamental difference, though, between that and gene editing, if you want to think about platforms across diseases. Because if you think of gene editing, you can kind of think of it as this modular therapeutic. You need an editor, which might be a messenger RNA encoding an editor. You need a delivery system. And then for CRISPR-Cas9, you need a guide RNA to tell the editor where to go. And the idea, the principle is, if you could show for a certain disease that this system works, now when you want to change the guide, or maybe even change the guide to a different gene and thereby change the disease, and you keep these two things exactly the same, that becomes a much more, I think, potentially efficient therapeutic platform. Because to develop a treatment for a new disease, you don't have to put another gene in another box. You just need a new guide RNA. And the guide RNA, the testing of those, the main concern of the guide RNA is to test for off-target editing. And that has to be done in human cells or human DNA. So no complicated, expensive animal models. So the idea here then, is we put as a third part of this some of these funding opportunities that basically required investigators to try to develop these platforms. They had to develop an approach that would involve one editor, one delivery system, and more than one disease. So we have one that's focused on Angelman syndrome and another similar autism spectrum disorder from Yale. They're actually using a chemically modified CRISPR enzyme that they can infuse directly into the brain. And then we have these two other programs, one focused on inborn errors of metabolism, and one focused on the inner ear, which again are designed to kind of do the same thing. Explicitly start as a therapeutic platform from the beginning. But then shortly after we funded this project, about a month after they started, something interesting happened at Children's Hospital of Pennsylvania. A little baby came in, was very sick, 2 days after birth, showing very severe signs. It was quickly determined that he had a form of urea cycle disorder. They measured ammonia, they realized what it was, and all of a sudden, although the project was not intended and doesn't fund clinical trials, we realized that maybe there's an opportunity to actually treat this person, this young baby. And it was a kind of interesting collaboration that came about, involved a lot of different parts of the SCEG Some private companies donated the editor and the delivery system. As I said, we were not able to fund the trial, but we did fund a lot of the stuff that went into the trial, the development of the drug, the animal studies. We actually gave them some additional money for animal toxicology studies that were required. And they were able, within about 7 months from birth, go to an actual treatment to this little baby. His name is KJ. The first treatment was about 7 months after, another one a few weeks after that, and the approval they got from FDA was to do 3 doses, each one higher than the last. And they ultimately gave him a 3rd dose, and he's gotten substantially better. His ammonia levels in the blood are much less. He was able to eat a normal diet, normal amounts of protein, which typically you would not do in a urea cycle disorder because the ammonia that is so toxic comes from the metabolism of proteins. And he's now home. So this turned out, again, we did not plan this one, we did not fund the trial, but it really gave the whole thing sort of a jump start as to how quickly this could potentially be done. And so what's moving now is that Karen and Becca are moving ahead with a pre-IND meeting, actually for their PKU trial, where they're going to be talking about treating 6 different PKU variants under some sort of umbrella protocol and seeing what the FDA thinks about that. We don't know. Obviously, there's been a lot of turnover at the FDA, so it's a little bit unpredictable. But I think there's a great opportunity here to sort of move this ahead in the liver. And the liver is a nice situation for this because we have this delivery system, and basically everything you put into the blood goes into the liver. So if we can sort of develop a regulatory path for a tissue where we have a delivery system, as we develop delivery systems for other tissues, we can kind of slide them into that regulatory path. This is the goal, this is the concept that we're thinking, obviously. We don't know if it's going to be successful. But speaking of better delivery systems, we did fund about 20 projects. We realized there's still some significant needs in the field, so we were able to, in phase 2 of the program, do something different, which is actually a prize competition as opposed to a grant. And a prize competition is just what it sounds like. People get money, they show us the data, and they get literally a prize. And we had one set focused on programmable delivery systems, where here they had to develop a way that you could target delivery systems to 3 different cells and tissues. And then the one that I was heading up is focused on something that I think is a big problem as a neuroscientist. I'm a neuroscientist by training, which is, wouldn't it be great if we had a non-viral delivery system that could cross the blood-brain barrier and edit cells throughout the brain? And so that's what this one is. You might ask, why non-viral? Ben Deverman from SEG's phase 1 has, in that program, actually developed a quite effective AAV that crosses the blood-brain barrier. So since he'd already done it, we didn't want to give him a prize for something he'd already done. He was a bit unhappy with that, but but actually he's using that same approach here. That he's a very smart guy. He's figured out how to identify the receptors for different AAVs and then put them into mice and use those mice as sort of human surrogates. So he's doing that for other tissues. So the thing about this is this is the second phase of getting $250,000. Ultimately, if they get the 3rd phase, they can get about another $650,000, but that's going to involve, again, we're going to need independent validation in large animals to show that this really works. And then just thinking a little bit farther down the line, you might think, I certainly thought, that if you had a therapeutic platform to treat a lot of monogenic diseases, that you could make $1 bazillion. Surprisingly, pharmaceutical companies don't see it that way. They think, understandably, in terms of developing one treatment for one disease, and then they can calculate a return on investment. which makes business sense. I'm not a business guy. But I think if we really want to treat as many people, we need something like a therapeutic platform, because otherwise no one's ever going to develop the treatments for those 1 in a million diseases. And so something that's interesting that they're starting now in the UK, and it was started by Tim Yu and Julia Vitarello. Some of you may know Julia because her daughter Mila was the baby for whom Tim Yu developed a personalized antisense oligonucleotide called Milasin. So they were really interested in the better regulatory path for the antisense oligos. Working with a former UK regulator named Dan O'Connor, in the UK they have something that they call the Rare Therapies Launchpad, which is kind of a pilot program for individualized medicine in the UK. And the idea here is that the regulatory system would essentially approve centers of excellence to do multiple treatments within a therapeutic modality, within certain specified constraints. And under that situation, they wouldn't have to come back to the regulatory agency every time they wanted to use a new oligo or any guide. And as they got the data, they would then report it back to the regulatory system. And the hope is that this might ultimately develop into something that can be reimbursed. So there would be a way to have a therapeutic platform that you can get reimbursement for. This is in the UK, of course. They have a very different medical system and different economy. So I don't know if this would even be possible in the United States. That's certainly well above my ability to think about. But I do think it's an interesting approach. That, again, they were thinking about antisense oligonucleotides, but in some ways I think genome editing along the lines of what is going on at CHOP and others would be almost even better. And then finally, earlier today, we were talking about newborn screening. And the idea that we were kind of thinking about is, well, if we're getting to approaches to treat monogenic disease that are really focused on treating monogenic disease as opposed to individual diseases, therapeutic platforms for monogenic diseases, wouldn't it be good if we had a platform to find all the patients that had genetic diseases? As opposed to now, the current system of newborn screening is focused on about 30 to 40 or so diseases for which there are approved therapies. So would it be possible to incorporate whole genome sequencing into the current state public health lab-based newborn screening program in the United States? And we first talked about this and discussed it with people who work in these programs. There were a variety of responses ranging from that's interesting to no. So what we wanted to do is essentially, and we're planning to do this, fund a feasibility study to see if it would be possible, what would be the impact of trying to incorporate whole genome sequencing into those state public health newborn screening programs. So we got money again from the Common Fund to do this, and we're in the process now of hopefully identifying a group who's going to carry this out. And so the idea that the way we're kind of envisioning it would be sort of a common sequencing and analysis program that different state public health labs would sort of interface with. And exactly how they would interface with that program, that's part of the feasibility study. You know, well, how would it impact their workflow? As many of you in this room know much better than me, these places are, they're not very well funded. People are working very hard. We do not want to make their lives a whole lot more complicated. At the same time, I think as we're seeing the ways that you could potentially develop treatments for different genetic diseases based only on the sequence of the cause of mutation, we need a better way to find these patients until they become symptomatic or before they become symptomatic. So this is the pilot project feasibility study, MBS by WGS, that we're in the process of launching hopefully very soon. And so I think I'm kind of going to wrap up here and just say that you often hear people talk about N of 1 treatments, ultra-rare diseases, nano-rare diseases. I don't know what a nano-rare disease is, and I try to avoid using any of those terms because I feel like they're focusing on the problem. And whereas I think the solutions are therapeutic platforms along the lines of what I've been telling you about, gene therapy, gene editing. And so that's really, at NCATS, at least in my programs, everything we do is somehow a platform. We don't do things one disease at a time. So happy to stop there and take any questions.
My goodness. That's really thought-provoking and very thought-provoking and a wonderful talk. This notion of, I grew up for many years following this, people spending their lives studying one disease to find one treatment for, and this is a paradigm shift that is a breath of fresh air, I think. But I want to pick up on your newborn screening stuff. I think the approach of testing newborns with whole genome for monogenic diseases is a really laudable one, but it's not what newborn screening was meant to do. And they do completely different things. Genotype predicts phenotype very poorly, and so I think doing genome in newborns would be great. Who's going to talk to the families about the results?
Yeah, these are all part of the feasibility study, whether it would be from some, maybe the people from the group that we fund could do it, or the local newborn screening team could do it with the appropriate training. Again, these are exactly the kind of questions that we're trying to address. And to your first point, I do want to I want to distinguish something. The goal of this really is newborn screening for very serious childhood diseases that could be potentially treated in children. We're using sequencing of the whole genome, but the plan is not to give people back their whole genome. The plan is to take from that platform really only and report back clearly actionable mutations from a small subset of genes for which something could be done. So we're using newborn screening to get all the data, but this is not about sequencing every newborn and giving them their whole genome, because that is a whole different thing, and you're right, that is not what we're trying to do here. We're just using, if there is some way to screen, for all these for diseases, serious diseases we could intervene in that was not whole genome sequencing, we would use that. But right now, for monogenic disease, that's what we have. So, one of the things that we'll also have to discuss is what genes are we going to look at. We only want to report results that are actionable. We don't want to be giving people back their BRCA1, BRCA2, because that's not newborn screening. That's providing their genotype, and that's not what we're trying to do. So I think, I'm glad you brought that up, and I want to clarify.
I think maybe a nomenclature change that would be helpful, because I think I read sort of newborn screening by whole genome sequencing.
Yeah.
And that everybody thinks means exactly what it says.
Well, I think
when you do a whole
means,
genome, you have to really.
But it means, I think the focus is newborn screening as we think of it now for serious diseases.
So you could exclude all adult onset disorders.
We plan to. Yeah.
You know that most newborn screening laboratories know next to nothing about calling variants.
Yes, and they will not have to. That's why.
So why would you do it how's that going to work then, do you think?
Well, the way we think it could work is that we will have some of the dried blood spots. They'll take some extra punches of those, send them to a sequencing center that serves multiple public health labs. We're envisioning 5 to 10. They will do the sequencing, they will get the variants, they will interpret them, and either they will go back and return the results, or they will work with the public health system to return the results. We do not expect that a big focus of what we're trying to do here is, and we've had a lot of discussion with public health labs, it's just how can we do this in a way that it's going to limit the burden on you, because we understand that they're already kind of, in a very difficult situation, so.
And in the first iteration of this is a research project, right? This is not clinical care work.
This is a research project. As such, it will have to be consented. And again, a big part of it is feasibility. Like, when you try to do this, what is it like? And I think there's been a lot of discussion and debate about this, about what people think is going to happen. But our view is we need to actually try it and find out if it's feasible or not. It may turn out not to be feasible, but that would be good to know. Or it may turn out that some parts are feasible and some parts are not. And again, if you're thinking about the potential of doing something like this in the future, that's the information we're trying to get. It is a feasibility study.
Yeah. Thanks a lot.
Thank you. Ronnie Singh from Emory University. Thank you for a wonderful talk. I'm going to not belabor this point, but want to expand, because as Propel and co-Propel grantees, when we were discussing the feasibility of this whole exome sequencing, one of the biggest issues is that many labs were not prepared or not had the knowledge of the background of the molecular part of the testing in the state labs. And yet the funding lacked the clinician follow-up, even if it was positive for the current diseases, if it came before the biochemical testing results, because with rapid genome, sometimes the results are coming in first. Like, who's going to report those? And currently, at least, majority of the lab in the region, when I talk with them, they didn't seem prepared. How are we handling that component of it in terms of your RFA?
Yeah, I think that we wrote it in a way that gave the people who responded, it was their job to propose how the return of results would be done. Because there is some experience with this in some labs, and we also encourage them, I'm sure a lot of them did, to talk to state public health labs. Some of them are already working with state public health labs to find out exactly how this would be possible without causing all these problems, realizing what knowledge is at the public health lab. That again is a real part of the feasibility study. I can't give you any details, because until we've actually funded it, and then people will see more.
Just quick second question. I was fascinated by the urea cycle story, because we all look optimistically and hopefully this would work, and then you jump to the PKU. Is there things like what Chuck did, with MMA, organic acidemias, where the outcomes are not good, you're funding from your other core to look at gene therapy, the AAV, would that be a potential population for that? Because those are the ones who could benefit from the script rather than the PKU.
Well, so Kieran Musanero actually has 2 grants. He has one on PKU. The second one is focused on inborn errors of metabolism, which would include organic acidemias and all those things. It just so happens that because his PKU grant was funded first, he's about a year ahead. But one thing, I think I skipped over it, unfortunately, on that slide, but one thing about the SCGE that is really, I think is really wonderful, is working out way better than we thought, is we have multiple groups here, but they all sort of work together when one of them goes to the FDA. So one group goes to the FDA, they ask some questions, we circulate that, then when the next group goes, they've learned what the first group did, and so we are kind of trying to do this in a sort of collaborative way that's so far working out really well, because everybody's working on other, multiple rare, there's no competition, in other words. So that's kind of how it's going.
Thank you so much.
But yeah, that would be great. Those are those are the most obvious ones, organic acid DNA stains you can mention, sure.
Other questions? Thank you so much. It's really great. Appreciate it.